And in this video, I'm going to explain about 9.2 about population ecology. So in the end of this video, there are two learning standards that we want to achieve. The first one is you as a student are able to describe the factors that affecting the population distributions. And the second one is we are able to analyze the data to estimate the population size of organisms using two techniques. The first one is quadrat sampling technique, and the second one is capture, mark, release, recapture technique. All right. So we got to the first one, the factors affecting population distribution. So we are looking at perspective of plants and also animals. So we start with plants. So what are the factors that maybe plants affects the plant's growth? Okay, there are five of them. So the first one is temperature, of course. So temperature optimum temperature for plant growth is about 25 degrees Celsius to 30 degrees Celsius. So that is the optimum temperature for enzyme in plants. Okay, when there's a high temperature, it will affect the plants because high temperature can cause the enzyme to be denatured. Okay, denaturednya asli. Okay, and this when that happen, the biochemical processes in plants will disrupted, right? And when that happen again, it's a chain reaction. It starts with high temperature, it will cause the enzyme to denature. After the enzyme denatured, it will cause the biochemical reactions in plants to be disrupted, and then the growth of the plants will be stunted. Okay, and of course, this high temperature can also increase the rate of water evaporation by transpiration. As you know, transpiration means a water loss process. All right, so we don't want that happen. We don't want our plants to lose the water. So because water is essential for plants, and of course, high temperature can cause to slow down the rate of the photosynthesis. So that what happen if the plants is around is in the high temperature area. Okay, it so much disadvantage. That is high temperature. How about low temperature? Low temperature can also affect um, the plants, okay, or the biochemical activities in that plant. So low temperature can reduce the activity of enzymes, so slowing down the biochemical reaction. So it will also slow down the reactions of photosynthesis. So it is important that plants it grows at the optimum temperature, which is 25 to 30 degree Celsius. Okay, so that is the first point, temperature. And water. Water is essential for plants because water is needed for enzyme activity, photosynthesis, of course, to transport, and also as a support for herbaceous plants. Okay, because when there's a dry region such as desert and tundra areas, such as at pole. Okay, tundra ni tempat yang sejuk lah, kutub. It has a low populations of plants. Kenapa uh, tidak ada banyak plants yang boleh hidup di kawasan yang kering, a drought areas, a drought region, dry region, or it's a too too cold or tundra. Okay, it's at poles di kawasan kutub, because there's a reason of that. There's a less of water available there. Okay, and light. Light, light energy is a source of um, light is a source of energy for photosynthesis to happen. So of course, we can make an observation. We can compare where the areas that receive sunlight exposure has much has high plant population compared to the shaded area, or compared to area that receive less sunlight, it has a low population of plants. All right. Of course, why plants needs this light? Because light is essential source of energy for photosynthesis. Okay, so far we have 3 points. Now we go to the fourth one, pH of soil. So um, pH of soil need to be in a neutral pH, because when the pH of the soil is too acidic or too alkaline, it will cause a lower absorption of nutrients. Right? So when there's a lower absorptions of nutrients in plants, it will disrupt the growth of the plants, because due to lack of nutrients. So plants need to be at a neutral pH of soils. So the fifth point is mineral salts content. So mineral salts content affects the absorptions of water through osmosis by the roots. So high salt content in the soil will cause the plants lose water through osmosis. Okay, we had learned this in halophytes. Okay, halophytes is where plants are um, able to survive in a surrounding where there's high salinity or high salt in soil. But usually other plants, they cannot survive because they need to be um, they need to be surrounded by low salts content. Okay, when the surrounding is too high salt, it will cause the water from the plants to diffuse out from the roots of the plant into the surrounding. Because of course, the surrounding has a high salt, aka at the same time it means the surrounding of or the soil has a low water potential. So when the surrounding or in the soil has a low water potential, in the roots of the plant, it has a high water potential. So movement of water is from high water potential towards the low water potential. So that is why the high salt content in the soil will cause the plants to lose water through osmosis. All right. However, minerals are needed for the productions of protein, enzyme, nucleotides, vitamin, and other compounds. Okay, as example, phosphorus is used to form phospholipids in the formations of cell membrane. Okay, so that are the five factors that affecting the population distribution for plants. The first one is temperature, water, light, pH of soil, and the fifth one is the mineral salt content. So, what are the factors affecting the population distributions of animals or other organism, or even us humans? So, in this point, we are talking about animals. There are four. Yes, there are 4 points. The first one is, of course, same like plants, organisms or animals. They need to be at the optimum temperature, not too high, not too low. All right. So areas which are exposed to high temperature in a habitat are less inhabited. All right. If you can see that there is no many species are available at the dry region such as deserts, right? And then even the tundra at the poles, there are not many species unless they have their own adaptation. But compared to the region that has really at the optimum temperature, we can find it more species that are available at there. Right? So water. Animals are concentrated in area that have enough water sources. All right, source for drinking and cooling the body, of course. So you can see there's water is also essential for plants, for animals, for such as that's for drinking and cooling the body. Okay, the third one is breeding site. Breed. Okay, breed here means to unable to produce offsprings. Breeding site is the location. So animals will look area that is suitable or safe for breeding site. Okay. So in order they produce and rise their offspring. So they are not being um uh they are not exposed to their predators and all that. So they're looking for areas that is safe and suitable to raise their offsprings, of course. So the fourth one is for food supply. So when the plant is looking for water resource, it's also looking for a area to live that have a food supply, of course. Why? Because food is important to survive, and as it's not that the area just available for what I want to say is that place must have available for plants that eat herbs and also other animals. All right. So, for examples, the animals that are heterotroph means animals they need to eat other organisms. So they need to find a place that have enough food supply. So whenever they find that their habitat is no longer have enough food supply, they're going to migrate or they're going to transfer to another place and looking for another place that has food supply. All right. So that are the factors that affecting animal population distribution. All right, the 4 points, which is temperature, water, breeding site, and food supply. All right, so next, now we go to the second learning standard that we're going to achieve is about what are the patterns of population distribution. There are three of them. The first one, clumped. Clump organism they will form a separated groups. Okay, in this condition is caused by uneven distribution of resource in an area. So in that area there are several groups will be formed depending. Let's say we have three clumps over here. At the first picture, you can see there's three major clumps, three major groups, because in this area there are where the um, factors good factors to be in that place. Let's say it has a good food supply, it has water, it is safe for breeding site, and the temperature is good. Right. Okay, random. So the second pattern for population distribution is random. So organisms are distributed in free pattern. There are no really a pattern that we can really look. There's a freely distribute. All right. This distribution involves species that have little interactions among individuals. Okay, maksudnya macam ada certain organism ataupun animal species that they prefer to stay in their groups. Macam kalau clump tadi, contoh um, the elephants, the lions, they stay in a group. Okay, but for this random pattern, it's a freely like some what it says. For example, the giraffe itself, okay, they tend to live without interaction. Dia tidak bergantung dalam kumpulan, so dia akan pergi sendiri. Alright, uniform. So uniform maksudnya ada pattern yang tersusun. Okay, organism are distributed all over an inhabited area. Distribution involves species in which every individuals competes for limited resources in an area. Okay, dia bersaing sesama sendiri. Sebab itu kenapa dia boleh ada nampak form dia. Okay, having said that, sometimes this is not just involve animals. This is also a pattern of plants. Okay, plants pun dia boleh ada bentuk population yang macam ni. Okay, so we go to the estimations of organism population size. So how do we say that that place has many populations of certain species? Let's say we go to our we don't really have a field at school. Okay, so we look at this field at this complex stadium complex kan. Kita mau tahu dalam padang tu dia ada spesies plant atau spesies rumput yang jenis apa banyak. Okay, so memang secara roughly we can see through naked kita boleh cakap okay jenis ini, tapi to have a structured data collection, kita ada data collection, there are two techniques that we're going to use. Okay, before that, what is a population size? Population size means the number of organisms. How many organisms in that species? All right. So there are another terms about population density. So population density is the number of individuals of a species per unit of a area of the habitat. Okay, density dengan size ni dia ada beza lah. Satu, from that population ada berapa species atau berapa organism, and kalau dalam density pula dia lebih kepada per unit area. Dia ambil sesetengah area saja dalam satu habitat. Okay, this depends on the habitat. Alright, so population size in an area can be estimated by random sampling technique. Okay, perkataan sampling. So meaning that because when the study area is large and difficult to obtain that data, bila kita mau tahu satu area tu ada berapa spesies and then berapa banyak spesies dalam area tu, and then area tu sangat besar, kita hanya ambil sampling. Sampling means we going for examples. Kita mau senang faham apa maksud perkataan sampling ni. Let's say we're going to do a survey. Kita mau buat survey tinjauan. So kita mau tahu apa pendapat what are the opinion of teenagers about this COVID-19. Contohnya lah. So kita tahu teenagers dalam Malaysia ni pun sangat ramai. So kita ambil sampling saja. Let's say kita ambil beberapa orang yang mewakili the voice of or the opinion of the teenagers. Macam itulah. Itu maksudnya sampling. Alright. So we're going to take certain areas to represent the population size. That is why we call it estimation of organism. Kita hanya buat anggaran. Alright. So there are two methods to estimate populations. Actually, there are many methods, but two of them is quadrat sampling technique, and the second one is capture, mark, release, recapture technique. It's such a long name for a technique, but it's actually tell you the whole process, what they actually do in this technique. So we're going to look at the first technique. What is quadrat sampling technique? All right, so quadrat sampling technique, we can use this technique to estimate the populations of plants or animal which are inactive or move slowly. Basically yang sangat lambat bergerak, macam snails lah, kita boleh guna teknik quadrat sampling ni. Kenapa? Sebab we prefer to use the technique sebab bila dia tidak bergerak, sebab kita kena kira dalam kuadrat. Okay, what is quadrat? Quadrat is actually the device that we use. Okay, you can see here there's a video. I already attached a video here. You can see that this person, they use a quadrat. Quadrat is a grid, usually in a form of square. All right. So okay, so you can see this what the person hold here is a quadrat, and what you can see that they used to count. Okay, let me pause that first. So and then let's say now this person wants to count the populations of daisy species. So they put this quadrat on the land and they count which grid that contain or which box that contain that daisy. Okay, itulah nampak dia macam pointing dia kira. So let's say dalam satu ni there are 5 times 5, there are 25 boxes grid there. So daripada 25 tu ada berapa box yang ada content that species that we look for, like the daisy, pokok bunga daisy. Alright. Okay, so this is the quadrat. The quadrat is a okay, so dia akan jarak setiap 2 meter dia akan letak, and then dia akan kira di mana yang ada daisy tu, and then dia akan record. Okay, so it's about the frequency, the density, and the coverage percentage. This is the what we call the formula to find frequency, of course, about the numbers of quadrat containing study species and over the total number of quadrats. Actually, we're going to need to do a hands-on like experiment activity for this in order to understand how do we use this formula. So far, for now, you just look at this formula. Nanti kita akan gunakan lah. Alright, that is the first technique, quadrat sampling technique, suitable for plants and also inactive animals yang tidak banyak pergerakan. Okay, right. So the second lah, dia ada video dari sini. Okay, so the second second technique. The second technique is about capture, mark, release, recapture technique. Sometimes is the shorter version of this technique, we call it capture and recapture. Kita tangkap dan kita tangkap semula. Kenapa? Okay, when I said the name itself tells you what is going on about this technique. First, we capture a species. Let's say we want to estimate the size populations of squirrel. So we capture squirrel. Of course, we put a trap. Kita letak satu perangkap lah. Cara kita tangkap. We capture and then we mark. Kita letak tanda. Okay, so masa kita letak tanda tu, make sure that tag that we use is non-toxic and it's not obvious, terlalu obvious sampai menyusahkan squirrel tu, sampai dia is being easy to detect by the predator ataupun sampai dia jadi susah untuk untuk mating. Okay, so tagging ni biasanya gunakan cat ataupun yang tersembunyi, kita letak tersembunyi. There are many types of the tagging kita boleh guna. Lagi advance, dia akan guna macam yang dia akan masukkan chip yang secara chip, jadi kita boleh detect bila secara ini tidak terlampau too expose. Okay, tidak terlampau ketara, too obvious. Okay, tangkap. Mark means kita guna tagging. And then lepas tangkap, kita tanda, kita release. Then release back to the habitat yang di mana tinggal. Kita biarkan. And then kita akan recapture balik. Okay, recapture ni bukannya kita aim, kita mesti dapat yang bertanda tu. Kita letak balik tempat yang di mana kita pasang perangkap tadi and then kita recapture. So by chance actually kita tidak bukannya naturally trap di situ. Maksudnya bukan kita aiming, kita mesti dapat yang bertanda. Just a recapture. So secara random. Jadi bahasa recapture tu kita tengok adakah kita dapat balik yang kita pernah tagging ataupun belum. Okay. At this point, nanti kita akan buat pengiraan lah. Okay. Masa kita tangkap, kita tanda dan kita release, kita akan bagi certain period of time. Maksudnya kita kasih biar dalam seminggu atau 2 minggu. Alright. Seminggu atau 2 minggu. A second capture is done random in the same area of study. Itulah second capture tu kita tangkap, bukannya kita aim dapat yang ada mark. Okay. And then the total number of animals captured of the second time, both marked and unmarked, is recorded. Maksudnya masa second time kita tangkap tu, kita bukan untuk kita tanda lagi, kita tengok berapa yang ditanda, berapa yang tiada tanda. Okay, from that kita akan buat pengiraan. Okay, capture, mark, release, recapture technique can estimate the population's size of organisms such as fox, sharks, snails, millipedes, insects, and butterflies, which can move freely in the studied area. Alright. So nanti actually sebelum kita pergi summary, dia ada perkiraan lah. From that data, what you can do with that data to estimate the size of the population. Itu pun sama juga, kita kena hands on baru kita tahu. Okay, jadi let's do, actually this is the end of the slides, tapi teacher ada satu lagi. I want to show you a virtual lab that has been established by Professor Jason, I think. We're going to look at it just in a second. Okay, so a summary for these slides, we had already looked at the factors that affecting population distribution, such as temperature, water, light, pH of the soils, and of course the mineral salts content. And for animals, the factors that affecting population distribution is temperature, water, breeding site, and food supply. Okay, so we had looked briefly the two techniques to estimate the population size. So the first one is quadrat sampling. We use a device which we call quadrat, and the second technique is capture, mark, release, recapture, or sometimes in other books they refer it as capture, recapture. Okay, so let me show you the virtual lab that I talked about just now. So actually this is the, yes, this is the virtual biology lab. Okay, virtual lab. So we are going to do this capture. All right. So we reset first. So let's say we have, this is the populations. Okay, so this is the populations of species. What species is this? It's an insect, right? So for instance, okay, this is the insect population that we want to count. So this is the habitat. We have 4 traps here. Traps number 1, number 2, number 3, and number 4. And then we place this trap randomly. Kita letak di mana-mana lah secara random. Okay, let's say number 1, I put it here. Trap number 2. Okay, put it a little bit on the right. Okay, and then trap number 3, put it at the corner here. Okay, and number 4, I think I'm going to put it up there. Okay, so the point that we place this trap is to catch, okay, capture, is to capture the organisms that we want to we want to estimate the size of populations. So there is a possibility other organism yang tidak berkaitan pula masuk dalam trap tu, it's okay. So kita lepaskanlah, tapi we only look at the organisms that we have interest with. Okay, so kita letak and then we open the trap. Okay, we open the trap. Okay, then there ada movement. Okay, I think I'm going to close my, sorry, I think I'm going to close my camera first. Alright, so look back. Okay, ada lagi laju lah. Okay, we leave this trap for sometime for instance overnight, satu malam kah ataupun 2 hari, seminggu, macam tu lah. Okay, so di sini dia close sudah. Actually we can control that, kita open dan kita close. Okay, lepas tu kita dapat berapa ekor yang kita dapat, kita kira lah. Let's say for trap number 1 we have how much is that? 3. Trap number 4 kita ada 1. Trap number 2 we have 2. Okay, then all that. Okay, so kita mark dulu, kita tanda dulu. Okay, kita tanda ni, dia guna warna merah pula, dia tanda. And then okay, itu tadi we capture, we mark, and then we release. Kita release. Maksudnya kita buka lah. Okay, sorry, release. Kita release. Kita release, so dia akan bergerak bebas balik di dalam habitat dia, environment. Okay, ini pun sama, kita bagi masa beberapa tempoh. Okay, maksudnya dia bergerak. Adakah organism ni dia tinggal di situ atau sebenarnya dia hanya berlalu saja. Jadi kita mau make sure dia betul-betul stay in that habitat. So after sometimes, maybe a week later before, macam tu, kita recapture. So kita buka. Okay, kita buka, kita mau capture balik. This is random. Kita jangan, sebab ini kita nampakkan. Tapi kalau ikut kita akan tinggalkan, ni dia close sudah. So ini adalah yang termasuk dalam apa. Kita boleh tengok sini. Kita boleh tengok sini dalam bawah ni. Total mark ialah 8 ekor. Lepas tu yang kita dapat kita tangkap semula in second capture, we have 5. Okay, we have 5. Okay. So ini nanti kita akan guna dia ada pengiraan dia lah, macam mana kita guna data tu, which is about how we use this data to estimate this size of population size. The formula is,
it's 8.
Okay, the population is like,
Mama, it was 8.
Yes. Okay, the population size is,
Mama,
the number of animals, the population, the number of animals in first capture. Let's say awal-awal kita dapat tadi berapa, I forgot to. Okay, inilah tadi lupa record kan, kita kena record data. Let's say we have tadi kita ada roughly ada 4, 1, 2, 4. 4 tambah 1, 5, 7, tambah 4, 12. Okay. So first capture kita dapat 12. Times with second capture. Second capture kita ada, sekarang kita ada 9. Okay, jadi 12 darab 9 over berapa yang bertanda pada second capture. Second capture yang bertanda adalah 5. So 12 times 9 over 5. Begitulah. Jadi jawapan dia itu adalah the size of the populations. Okay. So Itu sahaja untuk video kali ini. Kalau ada apa-apa soalan, nanti boleh tanya cikgu. Thank you very much for watching this video.